cycle, shorter incubation periods. Attachment. The phage attaches to the surface of the host. Penetration. The viral DNA enters the host cell. Biosynthesis. Phage DNA replicates and phage proteins are made. Maturation. New phage particles are assembled. Lysis. The cell lyses releasing to the newly made phages. Bacteria versus archaea. Cell size/cell type. Prokaryote. No nucleus and lack other organelles. Unicellular. Small single-celled organisms with simple structure. The cell shape is coccus, round-shaped, Bacillus, rod-shaped, Spirillum, spiral-shaped, Pelomorph, shapeless. Other shapes are pyramid-shaped and cube-shaped. Arrangement, strepto. Cells arranged in chain. Styplo, cells arranged in clusters. Diplo, cells arranged in pairs. Same for bacteria and archaea. Autotrophs, ability to make their own food. Photosynthetic, organisms that synthesize their own food with the help of bacteria-only light energy, sunlight. Chemosynthetic, obtain energy from organic or inorganic compounds such as bacteria and archaea organic fuel or hydrogen gas, respectively. Heterotrophs, need to take in food from an external source, can't synthesize their own food. Obtain their food from dead organic matter or live in the body of plants and animals, symbiotically or parasitically. Bacteria and archaea are both autotrophs and heterotrophs. Habitat. Bacteria tend to live in moderate environments referred to as mesophiles. Inhabit in almost every place on Earth. Archaea thrive in extreme environments, referred to as extremophiles. Methogenes live in anaerobic, no O2 environments, the depths of swamps and landfills, or intestines of cattle, utilizing CO2 and H2 gas to produce methane gas. Thermophiles live in extreme heat environments, hot springs and deep sea vents. Halophiles, salt lovers, live in saline salt environments, salt lakes. Acidophiles, acid lovers, live in extremely acidic environments. Thermoacidicophiles live in extremely hot and acidic environments. Pycerophiles live in unusually cold temperatures. Reproduction. Asexual, a form of reproduction which new offspring is produced by a single parent cell. Binary fusion. Asexual reproduction, a single parent cell splits into two identical individual cells. Produces a colony of genetically identical cells, does not allow prokaryotes to evolve to adapt to changing conditions despite errors in the copying process. Genetic recombination. Pieces of DNA are broken and transferred to a prokaryotic cell and mixed with their DNA. This creates new traits that may give new resistance, or the ability to adapt to changing environments. Conjugation. A transfer of genetic material from one prokaryotic cell to another. Gram staining. Gram staining uses a crystalline dye to show the difference in thickness of cell wall in bacteria and archaea. Only works for bacteria as it's a measure of peptoglycan. The more gram stain is absorbed, the thicker the cell wall is. The color appears purple/violet. The less gram stain is absorbed, the thinner the cell wall is. Appear red/pink. Gas exchange. The oxygen requirements of bacteria and archaea vary and can be used to classify them. Aerobe requires oxygen. Obligated anaerobe dies when exposed to oxygen. Facultative aerobe, using oxygen when present but can grow without it. Movement. Ciliated prokaryotes have tiny hair-like projections called cilia that work together in a coordinated rhythm to make them move. Flagella. Flagellated prokaryotes have long whipped tail projections that help them propel towards food. No specific structure for movement. You use a corkscrew motion to move. Glide on slimy mucosae secrete. Endosymbiosis. Endosymbiosis is a theory that eukaryotic organisms develop by combining two or more simpler cells, possibly prokaryotic-like ones. Evidence for endosymbiosis: mitochondria and chloroplasts, when compared to prokaryotes, revealed similar membranes, reproduced from binary fusion, each contains circular chromosomes, and are similar to day-to-day prokaryotes.